Again, guys, I'm telling, mitosis consists of 4 phases: prophase, metaphase, anaphase, and telophase. Now, I want you guys to have a look of how the cell looks like in the interphase, how the inside of the cell looks like. We have here something called chromatin, where the chromosomes are in this form. So we don't have chromosomes now. We have the DNA in a form of chromatin. Plus, we have structures called asters. They start to form in the interphase. It's actually centrosomes transforming into asters. And we have here something that covers the chromatin called the nuclear envelope or the nuclear membrane. Now, mitosis phase 1. What happens in the first phase of mitosis, which is prophase? First thing, guys, which you need to know, that we have here some fibers coming out of the asters. This is the first thing that happens. Number 2 is that we have chromosomes condensed. Finally, we can see now the chromosomes made up of sister chromatids, because the chromatin now condenses into chromosomes, so they are visible to you now. And number 3 is that this nuclear envelope or this nuclear membrane starts to break down. Okay, so now it's going to rupture. In phase 2, which is metaphase, spindle fibers will place the chromosomes in the middle of the cell, and this middle is called equatorial plate. Okay, so can you see, guys, here in the equatorial plate, the chromosomes are placed by the spindle fibers, which is the metaphase. In phase 3, which is anaphase, these spindle fibers start pulling the sister chromatids toward the opposite poles. It's like this. One chromosome, each chromatid is being pulled toward the opposite pole by the spindle fibers. So spindle fibers, as you can see, pulling sister chromatids toward opposite poles. And remember, pulling sister chromatids, not chromosomes. Mitosis phase 4, which is the telophase and the last phase, guys. Can you see here what's happening? There's a contractile ring. This ring is cleaving the cell into two. Okay, so now we have two daughter cells forming with the same number of chromosomes like the mother cell. And of course, chromosomes are of one chromatid, not two sister chromatids. So again, the nuclear envelope or the nuclear membrane will start to form. The aster will transform back into centrosomes. Okay, and we have contractile ring splitting into two daughter cells, splitting the cell into two daughter cells. Now, after the telophase, a process happens which splits the cell completely into two new cells. So can you see here, guys? We have this picture which happens in the animal cell, and we have here a picture from the plant cell. The difference is that in the animal cell, cleavage happens, but in the plant cell, there's something here happening, which is formation of the cell wall or the cell plate. Okay, and this is the difference between the animal cell and the plant cell. This is a final video which talks about all the phases together, or put all the phases together. As you see, guys, in prophase, the chromosomes become visible. And then we have here the spindle fiber starts placing the chromosomes on the equatorial plate, then pulling them in the anaphase toward opposite poles, pulling the sister chromatids. And then we have the telophase, which finally splits or cleaves the cell into two. Then we have the cytokinesis, which is the division of the cytoplasm, where two new cells are finally, or two new daughter cells are formed. Okay.